Um, Hello everyone, I'm Divyashree. So today I'm going to talk about how to design caps or decaps markers. I don't know how many of you are using these markers in reading programs or any genetics-based experiments. But I would like to talk about how you may not have to waste time on manually designing these primers, but use simple tools like Recaps Finder, where it gives you a lot of choices to choose either a forward primer or a reverse primer that you want to design for doing genotyping, especially like large populations like it could be either an outcross population or it could be a backcross population. So any type of population. Uh, Also, these decaps and caps markers are being extensively used in QTL mapping, quantitative trait loci mapping, because if you want to find quantitative genes, major genes controlling a specific trait, say plant growth or yield. So people use these caps or decaps markers for that as well, and also to construct the first genetic maps also, they would use caps or decaps markers. And widely, we use it for marker assisted breeding. So if you want to select a plant and you want to forward it to the next generation and use that plant for back crossing them into a stable line and to develop into a variety in the near future. So you could use caps or decaps markers to identify or to spot out where your gene of interest is present or not. And this could be detected in the form of SNPs or indels, insertions or deletions in specific regions of the gene of interest that you want to introgress into a population. Uh, Anybody have questions regarding this? And this could be a very basic thing. Many of you know about it. Uh, but I would start with a simple abbreviation.
Hello, Divya. Sorry to
Yeah.
interrupt you. There are queries like I think the voice is not clear. So can you keep your headset very closer to you and speak a bit louder?
Are you able to listen now? Are you able to listen now?
Yeah, I guess it's fine on my side. Um,
Okay.
Let me
Probably
ask.
I was a
Sorry,
little
is it
bit
fine
away
now?
from the screen. No, the internet is fine. There is no signal problem, but I think probably I was a little away from the screen.
Okay.
Maybe I have to push myself into the screen
Yeah,
to
yeah, yeah, yeah.
just talk. Okay. So CAPS means it's a cleaved amplified polymorphic sequence. So you can use these CAPS markers uh, to detect SNPs or which is single nucleotide polymorphic sites or indels, insertion or deletions in a gene of interest as either loss or gain of a restriction enzyme recognition site. So the first thing is I'm going to talk about how to design a CAPS marker using DCAPS Finder before going to the topic DCAPS marker design. So as I said that a CAPS marker requires an available restriction site, a restriction recognition sequence in the given gene sequence of interest. So that should be available associated to the SNP that causes the changes between a wild type and that causes the differences between a wild type and a mutant. Or I can say plant
Hello,
A
Divya.
or
Sorry to
plant B.
interrupt you. Hello, Divya. Since I'm a
Yeah?
mathematics guy, I would like to confirm some terms. Like, can you tell me what is caps marker, what is t-caps marker, and what is SNPs?
Okay, CAPS means it's cleaved amplified polymorphic sequence marker.
Okay.
DCAPS means it's a derived amplified polymorphic sequence marker.
Okay.
And the other one you asked is?
SNP, single nucleotide polymorphism.
It's a single nucleotide polymorphism. So you can find these SNPs, a lot of SNPs, different variations between closely related species and between two different genotypes in a particular species itself. Say A tomato and a B tomato, then you can find a lot of differences between them in the form of SNPs or indels located in the particular gene of interest, contributing to the specific trait. Okay. So here, and this is an example from my PhD project. So I was identifying candidate genes responsible for a particular phenotype in a population. So I did a whole genome sequencing and found out these candidate genes, and I found that they differ, and these candidate genes in the mutants, they differ by a single SNP, which is like, so if you compare wild type, that is your parent, your control, to your mutant, which has a truncated protein, so which is somehow different in the phenotype from the control. So this is differed by just a single SNP located in the gene of interest. So here I have mentioned, I have just pasted a small stretch of a sequence, and the parent sequence has a G, whereas the mutant, which has the altered phenotype, has the shift A. So I used an EMS, I induced the mutations to create a variety of mutants to study what kind of genes contribute to the specific phenotype. I hope it is clear if anybody have any doubts. So this is all about why I have pasted the small stretch of this sequence. So this is the first pair. So if you go to the second pair, this is the reverse complement sequence of the above pair. Okay. So the forward sequence in the wild type is the reverse complement, which you can see here in the bottom line, and which is applicable to the mutant as well. So for designing a CAPS marker, you don't need to specify the number of mismatches because you are looking for a restriction recognition sequence already, which is attached to the SNP of interest. So I would just typically paste this sequence. Like you can open the DCAPS Finder 2.0 using the above link. You don't need to download this. This is an internet tool. You can just copy and paste, say, this particular sequence here. And you can also paste the second sequence here. So the second sequence has only one difference, which is the SNP. And you can mention the mismatches as 0. And before going to submit and receive the output, and I would like to open the instructions for how to use the DCAPS Finder, which is fairly simple. So which is why I have specifically cut out only 50 base pairs in to operate this particular software and get the markers required. So the instructions are, it does say you have two haplotypes, say A and B. In our case, it is wild type and mutant. Okay. And the two sequences, it says it must be identical except for the SNP, because this is the same gene sequence. So I am only looking for detecting the SNPs. I just need to differentiate between my wild type and my mutant in a population, in a given population, which could be an F2 population or a backcross F2 population, whatever it is. It could be a segregating population, for example. So to differentiate between this wild type and mutant, so because I want to confirm that this is the gene that was responsible for the phenotype I verified in my mutant. So, and that is deferred by this small change in the small shift in the SNP. So I want to take 25 base pairs of the sequence before the SNP and 25 base pairs of the sequence after the SNP. So you cannot go more than 60 nucleotides to paste it in the box. And then for a decaps marker, you should enter the number of mismatches, which I will tell you later. And it also says you can only get one of the primers. Either you can get a forward primer or a reverse primer. You can get a forward primer for the wild type or a forward primer for the mutant, or a reverse primer for the wild type, or a reverse primer for the mutant. So the other pair, which you can easily design it manually from the gene sequence, and the overall product of the amplicon, that is the product that you will see clearly in a gel, in an agarose gel, should be between 200 to 300 nucleotides. And this is all about it. When you digest the product using restriction enzymes, like you can use either a 6 base pair cutter or a 4 base pair cutter or a 7 base pair cutter. So you will get all these options. What kind of restriction enzymes can be used, to get the to use them for digestion, as well as to obtain the output in a DNA gel, in a DNA agar gel. So this is the thing. But for CAPS marker, we will only be able to see the enzyme options which are based on the recognition sequence already available in the given gene sequence pasted. So this is the instructions here. So if you paste the wild type sequence at the top and the mutant sequence, and you just give 0 for mismatches, it is going to give you some options as output in a new tab. So let us wait for it. So I have already pasted the output of it in the second slide. So which is, you can easily see this. So this is the same thing I have pasted already. So the top pair is a wild type forward and the mutant forward sequence, which is used for designing forward primers. And then in the bottom line, you will have the wild type reverse and the mutant reverse sequence, which is the reverse complement sequence used for designing reverse primer. So unfortunately, I didn't find any matches for wild type forward and mutant forward and mutant reverse sequence. But I have an option for the wild type reverse sequence. So if you go back to the slide, so I have just highlighted in yellow. So the recognition sequence, which is seen in the wild type reverse complement sequence, is CGAAT. So the cutting pattern of this primer is, which is given in the next slide, where it cuts exactly here at C. So if it's going to cut at C, so I'm going to lose all of these base pairs in my product. So the next thing is you have to design. So since you have used wild type sequence for reverse primer, where you will see the reduced product in the wild type phenotype, you will have to design a forward primer from the gene sequence which you use to provide this particular stretch of the sequence which is pasted. So I'm saying the overall product size, which I would normally keep it as 150 to 200 base pairs. Once you use this HINF3 restriction enzyme, after doing a PCR, after you amplify a product, and then once you digest using this particular enzyme shown above, you will be able to see 150 minus 25 base pairs. You will only see 125 base pair product. Whereas the mutant, after digestion, show the uncut product. It will show the full amplicon, full size of the amplicon, which is 150 base pairs. So if you want to clearly see this difference, I have a gel picture, for example. Now you will not understand anything. I'm just opening it now. This is all like, I have not labeled anything, but this is just a gel picture which shows the representation of the cut and uncut products, what I'm referring to. So you have a plant, which is the mutant, and after digestion, it does not show any cut product. And then you have a wild type, which shows a small difference compared to the mutant. And then you have some intermediate phenotypes, which is called the intermediate between parent A and parent B, which is the heterozygote, which will show both the sizes, which will show both the products, I mean the product of A and B, because heterozygote will have copies of, will hold one copy of parent A and parent B, whereas homozygotes will have both the copies of the gene. And homozygote mutant will have two copies of the altered gene sequence, I mean with the altered SNPs. So this is a representation of how I would score a genotype in a population. So if I want a homozygote mutant, then I will choose the top right corner. If you see the second genotype, I will probably choose this plant to do further experiments on it. So if I want to introduce this particular mutant gene of interest into a population, then I would backcross it with the parent A, and I will try to introgress this mutant gene of interest into the population. And I will try to get a stable line in a segregating population later. So this is all about designing a CAPS marker. A DCAPS marker will also exhibit a similar pattern like this. But in case of a DCAPS marker, so I will close down this output actually. In case of a DCAPS marker, you will have to introduce mismatches to create a restriction enzyme sensitive polymorphism based on this target mutation. So based on the mismatches, you can have up to 3 mismatches. I would go with 1 and try to see if I get more options for restriction enzymes, or 2, or maximum I would go for 3 mismatches. So this mismatches will help to introduce or destroy a restriction enzyme recognition site. Right? So you will have to introduce these mismatches in the primer itself, so that you are creating a new restriction site, restriction recognition sequence site. Okay. So are you clear about it? If you have any questions, I think I'm running about. If you have any questions, please let me know before I go into the DCAPS marker. Hello?
So guys, any questions? So I have a question from my side, like I don't understand what is segregating population, and can you
Okay.
can you
So
explain?
segregating population is, so I would say an F1. So a cross between a parent A and a parent B will give you a first generation or a filial 1 generation. Okay. And this is a combination of A and B parent, and this will have one copy of both A and parent allele, parent genes. Okay. Or the gene of interest you are looking for. So if I'm going to just sow the seeds of the first generation, which is a mixture of both A and B DNA, if I'm going to sow it in the next generation, for example, I'm going to get different combinations of this alleles. So I will get, say, a 50% of the combination, a 50% of the population as that of the F1. So which is having both A and B parent copies. then I'll have 25% of parent A and parent B individuals in that population. So it will have
Divya,
AA,
are you explaining with
AB.
this gel picture?
Oh, sorry, probably I can explain this with the gel picture.
Yeah.
So, oh my God, sorry for the random picture.
It's okay.
So, so this is what I meant. So you have a parent A, which is having an uncut fragment, just one fragment, and a parent B with a cut fragment, which is what I'm showing here. And that is also, which is called, I would say, BB, a parent A having 2 copies of the gene AA, and parent B having 2 copies of altered gene fragment BB. And the heterozygotes will have both the copies of each of the parents.
Okay.
When you generate a population from this heterozygote parent, it will give a combination of what you see in a gel picture. You will see 50% of the population with having heterozygote heterozygote products, and you will have 25-25 with either parent A like or a parent B.
Okay.
So this is how you score a genotype in a population. So this is a tool where you can use these markers to select a particular plant which you want to go forward to the next generation. If you find, and you associate it with the phenotype which you are looking for, and you associating both the phenotype and the genotype, based on which you compare both, and then you select for the plant to go for further experiments, further breeding programs.
Okay, thank you. So, sir, I think Shiv has one more question. Shiv, do you have any questions here?
Yes. Can you hear me?
Yeah.
Yeah.
Okay. Divya, would you come to the slide, please?
Which one?
The program one.
Okay.
Yes. So you just mentioned that using this decap finder not O, we can design the primer only either for the forward sequence or for the reverse sequence.
Yes.
So what if we want a primer both like forward and the reverse then?
So you can design only one of the two pairs, because the other pair has to come from your gene sequence. This is also from your gene sequence, which is close to the SNP.
Let me put you
Okay.
example like this. For example, I'm working on one gene which cause methylation in the transcription factor, and then they do further regulation. So right now I have this wild type and I have mutant one, but I don't know in which one the mutation is there, and because of that this gene is working. So the exact motivation
Okay.
is I have to figure out the gene. Does the methylation is happening in the wild type or in the mutant one?
Okay.
So in that case,
So
my requirement is to design the primer for both mutant and wild type.
you could design the pairs for both actually using this
Okay.
tool.
Uh-huh.
But the thing is, one of the pairs actually come from your gene of interest, from the gene sequence. So, for example, let me open out. So, can you see this window?
Yeah, I do.
So, I have this gene sequence. Okay, so this is a gene sequence, and I found a mutation from G to A. Okay, and then I just selected 25 base pairs close to this SNP before and after for designing either a forward primer or a reverse primer. It could be for your wild type as well as for your mutant. So if you develop both for the wild type and the mutant, ideally it is going to give you a similar results. So either you want to show a restriction digested product or a reduced product in the wild type, or you should be able to see a reduced product in your mutant. For example, your methylation gene,
Yeah.
you should either be able to see a reduced product, like the product size in terms of base pairs in your wild type, or the mutant. So the other pair I would design, say, from here. I could design a forward or a reverse primer from here or here, so that it gives me the total product size of like 200 base pairs.
Okay.
Okay. I
Yeah.
hope it answers your question.
Yeah, it's clear. Thank you.
Okay. So if you are able to see this one clearly, so I have a sequence pasted here. So I would normally go to the TAIR, the Arabidopsis browser website, and then I would look for the sequence ID, and then I will copy the sequence and put it in a Word file, which I'll do it quite conventionally. And then I would select just 25 base pairs before and after the SNP that causes the altered phenotype in my mutant, and then I would paste it here. So I can get different combinations of either I can get a forward primer cut which shows a reduced product in the wild type, or it could show a reduced product in the mutant. Or I could design a reverse primer which could show a reduced product in the wild type or reduced product in the mutant. It could be anything. So then I will go for designing the other primer pair from my gene sequence itself, which should give me the total product as the total product size should be between 150 to 300 base pairs maximum. And I would basically run this to find a very vast difference. You know very well, you can go for a 3 to 4% agarose gel to differentiate this size difference. And then you can see in which of your, either your wild type or mutant, in which of your individuals you found that the methylation is altered. Okay. I hope you understood. Hello.
Yeah. One more question.
Yeah.
What if, for example, the sequence has a mutation on 2 spot?
Then you will not be able to find it.
I think I am facing the same problem. Okay. Anyway, thank you so much.
But in case of a CAPS marker, should be very cautious about having another restriction site in the same sequence. If there
Okay.
is another restriction site, then you will have like different projects. You will not be able to come out with a solid single project.
Yeah, I'm thinking that.
Then DCAPS marker will help you to destroy that second restriction site in your sequence. Because what DCAPS marker does it, we are actually introducing mismatches here. For example, let us go to the DCAPS marker for you to better understand how it can be even advantageous to CAPS markers. So in DCAPS marker, say for example, I have just pasted the same sequence. Actually, this particular sequence is good for developing DCAPS markers because I got a lot more options to choose what restriction enzymes I have. Like I can use the availability of such enzymes, and especially you have to make sure what are all the restriction enzymes available in your lab as well, and that could match the hits that you get in this software. Okay, So I'm trying to just add two mismatches, for example. So I'm just following what I have written in a PowerPoint presentation. So, and then I'm giving a submit, and then you can see the output here in a few seconds. So here as well, so I have all these the pairs of, I have the forward and the reverse pair for both mutant and wild type. And I have so many options here for, and so many options here when you create two mismatches in a primer sequence. So you can make use of it either in the wild type forward or mutant forward or wild type reverse or mutant reverse. So in my case, I think I chose mutant forward, for example, because I want to see the restriction digested product, the reduced product in my mutant. So hence I chose mutant forward sequence. And then I chose HPA. So the original sequence probably has GCA, APTC. But instead of that, because I introduced two mismatches, so the A is shifted to G and the C is shifted to A. And then what the restriction enzyme does it recognizes the sequence. It recognizes the sequence and it does a 6 base pair cut. So it cuts as GTT here. So what I will be able to see is, I will be able to see only, so the total product I have designed, the total product size is 157 base pairs, but I will only be able to see 133 base pairs in my mutant phenotype, in my mutant genotype, sorry. So because I'm going to lose this whole part of the sequence when you digest it. And similarly, and then I designed the other products. So because I used mutant forward sequence as the primer, as one of the primers, I went back to my sequence, the whole gene sequence, and designed the reverse primers. And then I used it for the PCR amplification, followed by digestion using HPA1. So similarly, you can use, say, a mutant reverse sequence. Now I want to develop a primer from the reverse sequence, and that will also show a cut product, a cut restriction digested product in my mutant only, not in my wild type. So then I went about designing the forward primer from the original gene sequence provided. So again, I used, say, MFE1 and then it's a 6 base pair cutter, and this will cut exactly here at C. It will restrict at C, and then I will be able to see only 120 base pairs out of the total product, which is 142 base pairs. So ideally, I will be able to see 142. So if you compare it in this gel, this is a 100 base pair ladder. So I will be able to see 142 base pairs for the wild type, but for the mutant, I will be able to see only 120 base pairs. So the 22 base pairs will be restricted by the enzyme MFE1. So this is how you can use this PCAPS finder to design different combination of markers you want. Any questions? Or if you find it difficult, I can always clarify it for you.
Hello, Divya. Thank you. I understood. Thank you. Thank you very much.
Okay. So, yeah.
So,
Anybody?
anybody else has questions?
So this is a very simple tool and it doesn't require any expertise, no command line, so you don't need to download it. So it's very quick to design as many primers you want. And you can always verify the melting temperature, whatever it is. So I usually go to say if I'm ordering the primers from Sigma Aldrich, so I go to the Sigma Aldrich and then I go to the oligos and then check the primers I want. So for example, you can just go to the oligos. You can order oligos, for example. I think it doesn't show me well here. But normally, if I want to order primers from, say, Sigma, then I would go to its Sigma tools and try to verify that these primers are all having a specific melting temperature, which should be like between 55 to 63 degrees, and 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 then I make sure that they meet out all the requirements as per Sigma, as required, as shown in the Sigma website, and then I would place an order to Sigma. So it's very easy, where you can just order loads and loads of decaps primers and then verify it with Sigma tools and then order it right away. So otherwise manually designing these takes a lot of time, and it's very difficult to do it as well. And you could also verify the restriction enzymes in the NEB website. So I usually go for New England Biolab's website, and then I would just want to make sure like what kind of restriction enzymes these are. Either these are a 6-base pair restriction cutters or a 4-base pair restriction cutters, to make sure that where exactly they would cause this slit, and to predict the expected product that you would get in your wild type and your mutant. So I would normally check, say, HPA1, and then you get the whole history about HPA1. So, and then you can see where it exactly cuts, and then based on which you can predict the product. You can also see what are all the requirements of the, what are all the buffers you need to use for your restriction digestion, and then and also would tell you like the temperature and the time you need to wait until the restriction digestion is over. So this is what I do to design primers, and this will give you an idea where, also it would give you an idea. You can cross-check with your catalogs, in your stocks in the lab, based on which you can use the specific restriction enzymes for designing these primers. So you don't have to like go for a primer, like a restriction enzyme, which is totally not available in your stock. You can make sure that you get at least 3 to 4 choices. So it is as simple as that.
So Divya, can we do all this function using primer 3?
You could. So for okay, that's a good question. So you could use you cannot use primer 3 for designing decaps. Probably you can use primer 3 plus for designing the other set of primer.
Okay.
That you want to design. Sorry, primer 3 plus.
Mm-hmm.
So you can paste the sequence here that you want, and then you can probably design one set of primer if you want. So say if you want just the right primer, the reverse primer, or a forward primer, you can just design it using the primer 3 plus. Just paste your sequence here, or you can copy the NCBI sequence ID or the TAIR sequence ID. Here I'm using all the references from Arabidopsis. So you can just paste it here, and then you can give your specifications, like what are all the regions should be included, like the sequence that should be included, and where it should target. So I don't generally use all these functions basically. I just pick left primer and pick right primer, and I just paste the sequence. And then I make sure, and then I shortlist the primer primes I want in general. So you can verify the melting temperature. Either these primer pairs would cause a secondary structure, or you have or any other parameters that needs to be satisfied for oligo preparation. So I would just cross-check it with Sigma Aldrich oligo tools.
Okay, so how do you prioritize these tools like decaps marker and primer 3 designing? Like based on which criteria you'll prioritize?
So decaps finder is a very specific tool just for designing caps and decaps markers.
Okay.
You cannot use, or you cannot paste a big chunk of sequence and design primers for molecular cloning, or to design primers to amplify a big chunk of a gene and put it in the binary vectors.
Okay.
So this is just exclusive for designing caps and decaps markers only. The primer 3+ can be extensively used for molecular cloning works.
Okay.
So if you want to clone a fragment of a gene, say 2 kb or 3 kb or whatever, into your binary vectors, and by making use of the multiple cloning sites, I mean the restriction sites, then you could probably use this primer 3+. It can come very handy.
Okay.
Or I would normally go for an offline app which is called Ape. I don't know how many of you know about this tool, Ape Primer Design. It's a plasmid editor. So, you get a big picture of the plasmid. You can put the plasmid you want. So you can download it. It is Windows based as well as OS based as well.
Mm-hmm.
So you can just download this tool. It's very simple. And here you see the snapshot. You can basically select a plasmid where you want to introduce your gene fragment into it. So you paste the gene fragments into the multiple cloning site where you want to clone. So you get a big idea, you get an overall idea or overall picture of how your gene fragment will be cloned into this plasmid. So based on which you can design forward and reverse primers to introduce these genes into these multiple cloning sites available in these plasmids. I mean, carrying the binary vectors.
Okay. So thank you.
So this will give you a whole picture. If anybody wants to use this software, I can always come back on some other day to talk about Ape as well.
Okay. Okay. So that will be very helpful, I guess. So Divya, you have one more question from Miss Bhavani. So
yes.
So Bhavani, you can ask your question directly. You can ask your question.
Hello.
Hello. Yeah. Hi, Bhavani.
Yeah, I'm Pavani. So, are there any other decaps like tools, like if yes, like how confident can I be?
Can you be a bit louder, or can you come a bit closer, please?
Yeah. So I asked whether there are any other decaps designing tools or like of the markers.
I mean, I just found this morning, like I have not been using it, but there is a tool which also, I mean,
Yeah,
which is also efficient. Which is also efficient in picking up caps markers, actually. So something called blast digester. But I have not seen this as like really available for use. I didn't find it very straightforward. But decaps finder it's decaps finder tool is a bit old as well, but still it has been very reliable too.
Okay.
It's very easy to pick it out, but there are so many. So I just found or read about this blast digester, but I haven't tried it.
Okay.
It does say that you have to purchase it. So it's not a free tool. So I would better go with decaps, because what it does is, if you want to go for a caps marker, and then you just don't select or add any mismatches to it. It does gives you the decision to make either to uh, get a caps marker or a decaps marker. So you just paste your sequence of interest.
Okay.
And this tool will decide either to go about with designing a caps marker, or if you don't get any suitable matches, then it automatically drives you to introduce mismatches to find a suitable decaps primer.
Okay.
So this gives you a decision either to develop a caps or a decaps marker. So
Yeah.
this is fairly simple, and it's freely available. So yeah,
Yeah. Thank you.
thank you. I hope it is clear. So if anybody wants to use the tools like APE or Primer 3 Plus,
Hi.
Hi,
Divya.
hi, yeah,
Yeah, can you explain me about that A primer designer?
APE.
Yeah, A primer designer. Can you explain one more?
Okay, I can do another demo on some other day, but I can just brief it out now for you. So this is just a tool where you give a simple download and you get a window like this. Okay, what I'm showing here. Okay,
Okay.
and here you can just paste the sequence of the binary vector you want to use for your cloning purposes, and also the gene sequence of interest which you want to clone into this particular vector.
Okay.
Okay, so you get all these sequences in linear format, or you can and you can also view it in circular format as well. But it would be highly beneficial to use in linear format to design your primers. So you can actually try to put this sequence exactly in between in the multiple cloning site you choose for your binary vector.
Okay.
So you exploit, say, two restriction enzymes in the multiple cloning site, and then you drop the sequence there. And then it will be easy for you to manually design or select primers flanking these sites. And then it will be helpful for you to amplify it and introduce it into the fragment. So while amplifying it, you will amplify both vector and the sequence, and then you will ligate it. So this will be useful for you to visualize where exactly you can clone your fragment into, and how best you can design your primers in the flanking regions. So I can demonstrate this in any of the slots coming in the next few weeks. So then it will be useful for you to follow up what I'm trying to say.
We'll definitely give you a slot for Primer 3 and this tool, Divya.
And APE. Okay.
Yeah, okay.
It's again a very simple and a free tool, and this is an offline tool, for example.
Okay.
So it will be useful for many people who are doing massive amount of molecular cloning projects.
Thank
Okay.
you. Thank you.
Okay, thank you.
So guys, any more questions? Hello.
Hello.
I guess there are no more questions.
I hope it is clear, and if you are finding difficult in any terminologies, or if I have been not clear about certain things, please voice.
Okay.
Anybody?
So I guess there are no more questions there. So maybe we can wind up.
Yeah, I understand it's clear.
Yeah.
So, or the other way around. Okay.
Okay. So guys, I hope everybody has understood the concepts and everything. So, but if you have any doubts, you can write mail to Divya personal. So in the beginning of the lecture, we have shared the email ID and the LinkedIn profile, so you can contact her in person. So thank you so much, Divya, for the wonderful hands-on session. So
Thank you, Mahesh.
yeah,
And I would encourage you to give me another slot for explaining APE as well.
yeah, definitely, I'll give you that.
Okay, thank you
Okay,
very much.
okay, thank you so much, Divya. So guys,
Thank you.
we are going to wind up the sessions, and we will upload the video in the Telegram platform. So thank you so much, guys. See you tomorrow.